Okay, so this next and final track of speakers is meant to sort of start to bring it all home. And this is this next speaker is a topic that I was introduced to, and the speaker I met at a discussion about constituents of emerging concern, focused primarily on recycled water. And this presentation, or a version of this presentation, was literally fascinating and kind of blew my mind a little bit about some of the ways that you can think about this stuff. So Mike Dennison is a professor at UC Davis College of Agriculture and Environmental Science. He grew up on the East Coast and went to Cornell to get his final degree, and is now a happy California resident transplant. So with that, thanks, Mike, for coming.
Thank you, and thanks for the opportunity to come talk to you today. What I want to talk to you about is a little bit different, but linked with the talks, and some of the things have actually interfaced nice with some of the previous presentations. What I'd like to talk to you about is the application of bioassays for environmental monitoring. And so when we think about taking samples, so we've got samples, we want to know what's in them. So those samples go through an extraction, cleanup procedure, and then they're subjected to one of a variety of different kinds of assays. So either they go into instrumental analysis, which is really the gold standard, where you can go in, you know exactly what you're looking for, you can measure the exact concentrations. There are immunoassay approaches where antibodies are developed against a particular chemical or closely related structures, and then bioassay approaches being mechanism of action or a toxic or biological endpoint used to detect chemicals that can affect that pathway. And each of these have advantages and disadvantages. Instrumental are great in that they let you know exactly what's there and how much is there. Limitations become instrumental's great for finding what you're looking for, but they don't help you find what you're not looking for, if that makes sense. Chemicals that can produce effects, but if you're not looking for them, you're not going to find them by instrumental, with the exception of new methods for non-target analysis, but that doesn't provide you with any toxicological or biological information. Immunoassays, I mentioned, are great for a given compound or structure-related compounds, but again, you don't know in that case exactly what's associated. So when we start to think about some of these, it's what chemicals are we measuring, knowns or unknowns, or if we're trying to find new types of compounds. The other is the measurement and screen. So the speed, the cost, the precision and accuracy of these. Instrumental analysis is great, but some methods cost you $1,000 to run one analysis. And if you've got 400 or 500 samples, not such a good thing. But you know exactly what you're getting. The other thing is that instrumental and immunoassays provide you no real information about the biological or toxicological activity of your sample. I'll talk about that aspect with a couple instrumental and bioassay in a second, but it also, those assays give you no information about a complex mixture, the interaction between chemicals in the mixture that can make things more active synergistically, additive, or actually can block the activity. So you may detect a compound in there that has no biological activity because it's inhibited. And the reason that this is important, and it's really hard to see here, but basically what this table is a compilation. It's a little out of date, but it's a compilation of 46,000 compounds. And basically, the different colors represent how much information is known. The green bar represents zero toxicological information about that class of compounds. And basically, the summary from all of this is that 46,000 out of 65,000 at that time, there was absolutely zero toxicological information. So when we're monitoring, we're looking for things that we know are a problem, but what about all of these other compounds? And this is 84. We've made huge numbers of more compounds since then that are in the environment. The same thing here. This is just another way of looking at it. The bar is indicating, the green bar is indicating toxicological information versus no information. So we know a lot about a little and a little about a lot. So what we want to be able to do is to be able to see the activity associated with the other compounds. If we look at complex mixtures, are there things that we really should be looking at that we're not currently looking at? Okay, so there are lots of different kinds of biological approaches for bioassays, many, many bioassays. One can look at acute toxicity, and so that's been described with some of the invertebrate assays or the fish bioassays. There are genotoxicity, endocrine disruption, neurotoxicity type assays that are out there, and there are many different kinds of formats. People can look at target enzymes. For example, acetylcholinesterase inhibition will give you measures potentially of organophosphates that are in samples. There are cell-based assays, signaling pathways one can look at, stress, oxidative stress. There's a whole series of these biological assays. But the point is that the bioassays can't be comprehensive because there are biological mechanisms that, one, are too complex to make a simple assay to measure an endpoint. But many of them actually are amenable to high-throughput screening approaches for monitoring purposes. And so really the issue becomes, what are characteristics that we need for a useful sort of bioassay? So I really kind of characterize them into two sort of categories. There are bioassays that are really chemical and chemical class specific. And I'll talk about one of those. And a few of these are really targeted with a cleanup procedure targeted to a very specific class of compounds. And in this case, a cell bioassay looking for dioxins, like compounds, dibenzofurans, and PCBs can be very specific. However, most bioassays are really open-ended. It's biological endpoint, and you're looking at whatever chemical or chemicals in a mixture can affect that endpoint. This becomes important because when we think about, well, what's a relevant endpoint? Well, we think about AOP, so an adverse outcome pathway. So where we're looking at a biological response that is a critical step in a pathway that can lead to an adverse health effect. And so we would look at toxicity pathway, mode of action. Adverse outcome pathway goes from the chemical properties all the way up to population level effects. Where do we work with the bioassays? The bioassays focus in this region, right, which would be biochemical, cell-based, the critical steps in that pathway that can lead to alterations in population. All right, secondly, if one has a bioassay, that bioassay really needs to be confirmed directly related to the adverse outcome pathway, and that would be concentration response or dose response relationships between concentration, increased response of that pathway, and increased adverse effects if you're having an optimal bioassay. The other thing becomes an issue of toxicokinetic analysis. So when we are using these bioassays, they are in vitro. We have given concentrations. If we see an effect, what's its relevance to in vivo? Well, what we need to know is what would be, for example, blood levels of chemicals. How does that compare to the concentration necessary to produce a response in a bioassay? Is it comparable to blood levels? Then one would expect to see similar effects in the in vivo situation. The other issue is that we're looking at chemicals who work through a common pathway or a common adverse outcome pathway or mechanism. Okay, so I'm going to go through a couple of these. And I'm not going to go through lots in detail, but the first one, as I mentioned, is a chemical-specific bioassay. And this really targets dioxin and dioxin-like compounds. These compounds are ubiquitously distributed, produce a wide range of species and tissue-specific toxic and biological effects. And they're distributed everywhere. Everyone in the room has a little bit of halogenated aromatic hydrocarbons in them. You could consider that maybe it's an essential nutrient. That's why we all have them, not a contaminant. So where do they come from? Basically, they come from any combustion reaction. Some of the early identification was herbicide spraying because of a contaminant in Agent Orange. The PCBs and transformers, which estimated that 50% of that is in the environment. You find lots of contamination in sediment, waterways, soil, food, and that's our primary source of exposure, is through food. The gold standard for measurement of these compounds is high-resolution GCMS. Samples come in, they undergo a cleanup to get rid of the unwanted compounds, focus on these. They go into instrumental analysis and allow you to calculate the exact concentration based on standards of the specific compounds that need to be regulated. And so you can get an exact measurement. However, it's an instrumental analysis measure that tells you how much is there. It doesn't tell you the biological response. What is done from an international perspective, and also in the US, is the use of toxic equivalency factors. So of the chlorinated dioxins, dibenzofurans, and PCBs, as you can see, there's more than 70 polychlorinated dioxins, more than 100 dibenzofurans, and more than 200 different PCBs. However, a very small number actually produce toxicity by this common mechanism. And in vivo studies have allowed calculation of their relative toxic potency. So if we look at what we would refer to as dioxin, the most potent, given a factor of 1, and everything else is expressed relative to that. So if you think about it, if you have instrumental analysis, you know what the concentration is, you can multiply that times its toxic potency and do that for each one of the compounds and simply add them together and get an overall toxic equivalency of a sample. And that actually works quite well in terms of estimates of potency. And this is used regulation-wise internationally for looking at this complex mixture. And the reason that that works is that all of those compounds produce their effect through the same exact mechanism. It's through a receptor, a dioxin receptor, an H receptor that's present in cells. If you get rid of that receptor, you have absolutely no response to dioxin. So it's what's responsible for producing those effects. What that receptor does is if we look at a dioxin responsive cell, these chemicals get in, bind to this receptor. This receptor migrates from the soluble fraction into the nucleus of the cell. So it works very much like hormone receptors, estrogen receptor, for example. Binds to a very specific sequence of DNA, induces gene expression, and that gene expression is what contributes to the toxicity. So we have a mechanism, and every one of those toxic dioxin, furans, and PCBs work by a common mechanism. So what we've done is to actually engineer cells where we've taken this DNA binding sequence for the receptor, put it next to the gene for firefly luciferase. That's the enzyme in fireflies that produces light. So we've engineered cells that have this permanently integrated into their DNA. So what happens? Expose them to dioxin or dioxin-like chemicals. This pathway gets activated. You produce more firefly luciferase, more light, more luciferase, greater concentration of compound. So how the assay works in our hands is a 96 well plate. You put these cells in the plate, add the compound in an extract to them, let them sit 24 hours. You lyse the cells, they go into a microplate luminometer, and it quantitates the amount of light output. And so the top line here is a dose response with dioxin. The triplicate samples would represent triplicates of different extracts. The differences in light intensity are indicative of the concentration of dioxin-like compounds in the sample. So what one simply does is get information from each of those, and you can get a relative potency value. As an example, this is luciferase activity, light production versus chemical concentrations, dioxins, dibenzofurans, PCBs. So those relative potency values that I showed you are exactly the same for the cells. Why? Because both of them are mediated by the same exact receptor. So you could use these cells then to do screening. So minimum detection limits with the current system we have is about 50 parts per quadrillion of dioxin for detection. Basically how the assay works, samples would come in as before, get cleaned up, they go into the cells, you treat the cells, you measure the activity, and then one can get a relative equivalency factor like we would do with the instrumental analysis and be able to do comparisons. The one thing about bioassays that's really important is false negatives, right? Because you're not measuring an individual compound, you're measuring a biological response. So what if there is something in there that actually inhibits that response? You'll say this is negative. Cell bioassays have the advantage in that you can eliminate that false negative. And basically what you would do would be to take a sample that's negative, add a certain concentration of dioxin that should give you a certain amount of light units, add your sample to it, and if it reduces the number of light units, it's having an adverse effect on the pathway or toxicity of the cells. And it allows you to eliminate then false negatives or identify true negatives, at least below the level of detection. And so with that, if you do have a positive activity, what do you do with that? Well, currently, Belgium is using this for food screening, and they have a trigger value, a cutoff. If activity is over a certain point, it immediately goes for instrumental analysis. If activity is below it and not a false negative, they don't worry about it. And so they're using it as basically a monitoring. So this is an example of double-blinded analysis with this bioassay for dioxin-like compounds, a comparison of, at least this is some of them, but it was about 250 real-world samples, so not standards, that went into the cell bioassay for equivalent calculations, and it was also sent off to another group for toxic equivalency by instrumental analysis, and what you can see is a really nice correlation between the ability to activate the cells and their toxic potency, which is not, again, not surprising because it's the same mechanism. This assay actually has been approved by US EPA as method 4435 for screening for a variety of environmental matrices. The one thing I do want to point out is that you can see there's two lines here, XY equal 1-to-1 between instrumental analysis and the cell bioassay. That occurs with biological samples. What we find with environmental samples, and we've done a number of water samples that are down here, is that the cell bioassay tends to pick up more activity than instrumental says that's there. So people for a long time argued that it's not a very good assay. Well, again, if you think about what is instrumental measure, it lets you look for what you want to find. Chlorinated dioxins, furans, PCBs. Well, these samples also contain brominated dioxins, dibenzofurans, and brominated biphenyls, polychlorinated naphthalenes, other things that can actually work and produce the same spectrum of responses. So it's actually been used to identify other types of compounds. So one of the things that we have done has been working with people over a number of years, and so this is a study that we did with people at SQUIRP, looking at 31 estuarine samples from Southern California where they're taking water samples, extracting them. They're running several bioassays. We were running the H receptor-based bioassay, and what, hard to see here so far away. So we looked at basically agriculture, urban, and then open areas that they had categorized based on use. And what we're looking at here is activity. So red would be activity in the assay, not quantitative, but as a percent of dioxin, everything over 30%. So we had some samples that were giving us 50% of that of dioxin, down to green where it was low and blue was basically nothing. So what we've been able to do is to look at this whole series and get a relative activity in each one of these water samples. And then what was done was to characterize this with regards to this CSCI, or the bug index, which I like. And what they can see is that there's a relatively good correlation between a decrease in the health of the ecosystem with an increase in activity of what we detect with the assay. Now, that doesn't mean the chemicals that activate the assay are responsible for that, but they may be a contributor or it may be just coincidental with whatever else is coming into that pathway. So currently, they've identified a variety of flame retardants and are still doing some instrumental analysis. So this assay has been used for lots of different types of matrices. Lots of publications have come out on this. The assay works because it's a cleaned up sample and you know everything's working by the same mechanism, and you have a very defined class of chemicals based on that extraction. So you know that you can make some conclusions. But it's not true with all bioassays. So as I mentioned, there are other bioassays that are more open-ended. They're not targeted to one class of chemicals. The other bioassay I'll mention is that for endocrine disruptors or endocrine active, or in our case, estrogenic or anti-estrogenic chemicals. Right now, we know about chemicals that can activate. How many people here have gotten rid of plastic bottles because there's BPA? So the concern about BPAs being estrogenic, but we don't know what the suite of estrogenic chemicals are. In fact, there are a lot more out there that are far worse than BPA. These compounds produce a variety of effects. I have some of the compounds indicated here. Where do they come from? You find them in the environment from lots of places, from pharmaceuticals, flame retardants, pesticides, sunscreens, plastic products. Dioxins are potent anti-estrogenic compounds. You find lots of these kind of chemicals, and so there are lots of exposure to these from a variety of sources. The estrogen response is a system very similar to that of the AH receptor response. It's built off the estrogen receptor. And so the mechanism is very similar. Estrogen compounds come in, estrogen binds to DNA, induces gene expression. So we can engineer cells such that the luciferase gene is increased in response to estrogenic chemicals. So we've made cell lines. We have human ovarian cell lines that have this system in. If you expose them to estrogen or estrogenic chemicals, you induce activity and you get an estrogenic response and increase in light production. Extraction preparation is basically the same, except again, it's dependent on what types of chemicals. Usually if we're looking at estrogenic, you're looking at more phenolic, more water soluble. Go into the bioassay. Positives can be used for calculation. The issue, as I mentioned, is we're not focused on individual compounds, so it's still somewhat open-ended as to exactly what the chemicals are, but one can utilize the bioassay as an approach to get rid of false negatives, just like we would do with the H-assay. So you have a way to get to positives as a way to get to negatives. We've done lots of screening with this, and so we've monitored and measured lots of different pesticides, for example, and you can see this is estrogen, and lots of organochlorine pesticides. And they're active, but they're really weak. In fact, BPA would be over here as well. Not very weak at all. So you'd have to have pretty substantial concentrations. The other thing is that we've looked at lots of plastic and rubber products. Really so hard to see in here, but we've done a variety of plastic bags, boxes, milk jugs, everything that we could find in the lab or at home that had plastic we extracted. The interesting point here, so the first bar is estrogen in both cases is extracted with ethanol or DMSO, in which you can see it's the same products, but you see different patterns. Different solvents are taking out different estrogenic chemicals out of those materials. The one thing I do want to point out is just because something says it's BPA-free doesn't mean it's estrogen-free. So we've analyzed a whole bunch of BPA-free polycarbonate, and it's full of estrogenic compounds. It's just not BPA. So if it's free of estrogen activity, the other question is it an endocrine disruptor? So we hear about estrogenic, and then we hear about everything's an endocrine disruptor. In our view, it's endocrine active in the bioassay. An endocrine disruptor produces an adverse effect in vivo, two different things potentially, right? You can have something metabolized extremely fast in an organism and not so much in the cells. So that becomes an issue with regards to the bioassays. We have done a variety of these. Looking at water, so everybody always has this question about the water jugs and the water bottles. What we did was basically analyze the water that was in here, doing this for this one group. With analyzing all 500 mils of water, we got absolutely nothing in this assay. Even with the polycarbonate, because bisphenol A is not soluble in water, it's not going to come out of polycarbonate into water. But if we take each of these and extract them with ethanol, the 4 bottles are here, they basically have nothing. That's the carboy. So that's the polycarbonate. Lots of BPA comes out of it with ethanol. So if you store your ethanol in a 5-gallon polycarbonate, that's probably not a good thing. Okay? But in general, it's not going to come out in water. And what does come out is going to be really trivial low levels. So we have done some environmental screening with these. So this is samples that we had collected at the Mokelumne River, the 3 tributaries of the Mokelumne River. We got 1 liter samples, filtered them to get rid of particulate, and then extracted the water, because what we're looking for is what's dissolved in water. And these are the results. And so this is luciferase activity. It's a percent of estrogen. Our method blank, these represent 4 different locations. This is 100%. So we actually have several of these samples that are giving us 80% of estrogen in our bioassay. And in fact, activity was found in each tributary of the Mokelumne. And we found it at 4 different times of the year. For those people that are interested in concentrations, they worked out to be, in this case, this is activity in the equivalent of 10 milliliters of water. So it's almost activating this fully. Works out to about 80 picograms of estrogen per liter of water, which is huge. The issue here is that we don't know what the chemical is yet, or what's the responsible compound, but it was present in each of these. And we've talked to several people about doing what's called effect-directed analysis. We can use our bioassay and coupled with people that do instrumental analysis to fractionate samples, use the bioassay to look at the fractions and work our way down to try to identify what the responsible chemicals are. But in this case, we don't know what that activity is. The last thing I'll touch on is that the bioassays that I'm talking about, as well as a number of other bioassays, have been analyzed by a variety of groups. So to Bioassay, they used 103 different bioassays. And lots of them that were the same targets, but they looked at a variety that were targeted to metabolic pathways, oxidative stress pathways, genotoxic pathways, and they looked at water samples. So drinking water samples, water reuse, gray water, and so on, to look at relative activities with the idea that they might be able to utilize these bioassays in longer purposes. And so what ultimately came out, these were the water samples they tested. Each of these samples of water had different characteristic profiles based on the types of bioassays that were affected. Most of them are related to xenobiotic metabolism, hormone receptor pathways, genotoxicity, and oxidative stress. And so what that allows us to do is start to focus on those as being more of an optimal bioassay combination. The other thing that they wanted to do with this, and they have done in separate papers, is to talk about using these bioassays as basically setting benchmarks or trigger values in terms of you run these, and if activity is over a certain point, then one goes on to either further analysis or targets these as a potential concern. So the conclusions are the bioassays have a lot of utility, but I'll be the first one to say they're not a replacement for instrumental analysis. They allow you to find an activity and then in combination allow you to work with instrumental analysis to say, here's 500 samples we just ran. Look at these 20 and put your money into the analysis of these 20 to figure out what the issue is. Critical things are extraction method, because when we look at the estrogenic, are you looking at hydrophobic, hydrophilic compounds? That's going to affect how you're going to extract things. What are the identity of the compounds? The toxic potential in vitro. And one of the key things that's been talked about for the last couple of years, there's meetings that I've been going to, is a critical interpretation of the results. If we run a bioassay on a water sample and it's active in an estrogen assay, what do we tell the general public? If we're going to stop this, we say, well, this is going to cause harm or not cause harm. This becomes the conundrum. And so people are trying to work right now to determine what characteristics, what things you need to be able to say that makes these assays really useful. But really, I think the important thing is that they're great for monitoring purposes. At this point, I would say not for regulatory purposes. You'd use them to do screening to help you in terms of identifying new compounds or current compounds. And with that, I just like the balancing act between bioassays, their utility. So, thank you.
So we have time for a couple questions. Anyone? People's minds got blown too much. Flonase worse than anything else I expose myself to? No. Okay. Just checking.
Short one. How did you pick the McComney River for your field study?
There was a group of people that came to Davis looking for people. It was a collection of organic farmers, and they were concerned about potential regulation with endocrine disruptors, and they were getting their water for their farms out of the McCollum. And so their concern was, what if there's stuff in the water? And so when we did the assays, it was a lot. We've never figured out where it's coming from because the samples are so far up. There's no pastures there. There's no houses there. So could be synthetic, could be natural. We do find activity in what sediment you can find at the collection site. Usually it's mostly rock because the water's going so fast. But that's where it came from. People came to us to say, could we take a look at it?
We've been working on cyanotoxins, and they show up in all these high watershed locations. You don't expect them. And I just decided to Google are cyanotoxins estrogenic and came up with an article. So it might be something to look at.
Yeah, we did. Look at this. So she said they're finding cyanotoxins. So it's algae. I know everything's algae, right? So that they're in there, but are those things estrogenic? So you find those in the waterways, right? So it could be, we could argue they could be phytoestrogens, they could be things from plants. Some people had argued with us that it's from snowpack. When the snow's coming down, it's pulling contaminants out of the atmosphere, depositing it when everything runs down. At this point, we don't know, but what we do know is that it's not affiliated with particulates because they go through a small micron filter. We just want what's soluble. So it's not the real hydrophobic stuff, and it'd have to be pretty small stuff to come through. So at this point, we don't know.